Hi everybody, my name is Ummat Somjee and today I'll be presenting Anti-nausea for Darwin, the energetic costs of exaggerated sexually selected traits. Sexual selection drives the evolution of a dizzying diversity of traits, from the horns of rhinos to the horns of horned beetles, from the antlers of elk to the tiny antlers of antlered flies, from the massive tusks of elephants to the miniature tusks of tusked weevils, from the ornamented plumage of many birds, and even the feather-like ornaments of canopy-dwelling mosquitoes. The existence of these sometimes extravagant and opulent traits was a point of consternation for Darwin. The year after The Origin of Species was published, he wrote in a letter, "The sight of a feather in a peacock's tail whenever I gaze at it makes me sick." Of course, many of us know that he later dedicated an entire book to this subject of sexual selection, where he proposed that competition for mating opportunities can lead to the evolution of many of these exaggerated structures. But in some ways, the question still remains. Why can some animals afford this exaggerated morphology and others cannot? We know there must be upper limits to the exaggeration of these structures, but what determines these limits? This question about upper limits gets even more interesting because many animal weapons exhibit positive allometry. This is what a male Hercules beetle would look like if its horns scaled in direct proportion to body size as it got larger. In this case, a large individual is simply a scaled-up version of a small one. But this is not what we see in nature. Instead, this is what we see. As animals get larger, sexually selected traits often get disproportionately larger. This highlights the conceptual question: how and why can large individuals invest disproportionately more in these structures compared to small ones? Two major explanations have been larger individuals may be able to invest in larger traits because they are better in some metric of condition, or that relatively larger traits in large individuals may more accurately signal high competitive ability or quality. While these perspectives may shed light on why individuals of the same species invest disproportionately more in sexually selected traits, they do not necessarily explain why positive allometry occurs across species. The smallest deer species we know is about the size of a small dog and has tiny pointed antlers that account for about 1/10 of 1% of body mass. While one of the largest deer species we know to have ever existed, the extinct Irish elk, had massive flaring antlers that accounted for more than 10% of body mass. Across species of cervids, large species invest disproportionately more in antlers. And a remarkably similar pattern is found across species of bovids, which have horns instead of antlers, a very different structure. So why is it that larger species invest disproportionately more in sexually selected traits compared to smaller species? How can these large species afford the apparent increased costs to bear disproportionately large structures compared to small ones? But there is another well-known trend that is likely linked to body size. As animals get larger in size, their bodies use less energy per gram of tissue. This has famously been called the mouse-to-elephant curve. Larger animals use energy at a lower rate per gram than small animals. There are many predicted reasons for this finding, but for this presentation, I would like to focus on just one of these. The system composition hypothesis suggests that as animals get larger, they need to dedicate more resources to structural support. These supportive structures are typically relatively inert. So while they do contribute to mass, they do not contribute as much to metabolic rate, resulting in lower scaling of metabolic rate with body size. A mouse, for example, dedicates only about 5% of its mass to its skeleton, while an elephant dedicates more than 20% of its mass to this metabolically inert skeleton. When we examine allometry, or how different traits scale with body size, we will often focus on the size of the trait, but not on the amount of energy that trait consumes, and this may be misleading. For example, we know that the human brain accounts for only about 2% of body mass, but accounts for about 20% of daily energy needs. So how does energy scale with sexually selected trait size? I first applied this energetic perspective to the heliconia bugs, where males invest in large hind legs modified as weapons in competition for mating opportunities. In this species, males exhibit positive allometry of weapons. Large males invest relatively more in weapons compared to small ones. We found that the energetic costs of these traits do not scale directly with their size. Large males pay lower costs per gram of tissue to carry these large structures. What are these costs, though? When we look into these traits, we see that males have high muscle mass and high tracheal capacity. We also see high mitochondrial activity in these muscles, suggestive of high respiratory capacity. But we really don't know how the proportion of metabolically inactive, trachea and cuticle, and active, nerves, muscles, and tendons, scales relative to each other as these traits get bigger. To explore how weapon composition changes with body size, I studied the New Zealand giraffe weevil, where males carry this elongated rostrum used in jousting-like competition for mating opportunities. Males vary remarkably more than 30-fold in body mass, and large males bear disproportionately large weapons. When we look into the structure of the weapon, we see that it's composed of metabolically active muscles, nerves, and tendons, surrounded by relatively metabolically inert cuticle. In fact, we found that large males carried larger weapons for lower metabolic cost, while small males dedicate about half their weapon mass to expensive metabolically active tissue. Large males dedicate only about 1/5 of their weapon mass to expensive tissue. When we typically look at sexually selected traits, we often focus on measuring the size of the trait itself. We know, for example, that larger species of elephant carry disproportionately large tusks. However, we also know that these traits are often highly integrated with other morphology to support their form and function. For example, a recent paper showed that not only do tusks of elephants scale with positive allometry, but that there's positive allometry of the head muscles in elephants, and the biggest elephant species likely had extra muscles to support their giant heads and disproportionately large tusks. In fact, many of the sexually selected traits that we measure are composed of relatively metabolically inert tissue. For example, the keratin in the ornamented plumage of birds or the horns of bovids and rhinos, or the bony antlers of cervids and ossicoins in giraffoids, or the dentine found in elephant tusks, or the modified cuticle found in many arthropod species. In fact, these structures may be prevalent in certain groups not because they are costly, but because certain phyla have found metabolically cheap avenues to sustain them. We know weapons, for example, are found in diverse groups, but at a broad phylogenetic level, how common are weapons and ornaments in nature? In fact, weapons and ornaments, as we define them, are found in less than 12% of phyla. So going back to the question, how can animals bear the cost of these exaggerated structures? Because these traits are large and draw our attention, we might mistakenly assume they're more common than they are. At least at a broad level, across phyla, they're not very common. Few animals that do have these traits capitalize on cost-saving physiological mechanisms to reduce these costs. Finally, direct costs such as increased predation due to large traits have been surprisingly difficult to document. Finally, I'd like to bring this back to Darwin's fascination with the peacock, which has become a poster child of sexual selection. Recent studies on peacocks specifically have revealed that peacocks' trains surprisingly do not impede takeoff performance, and peacocks with trains had lower, not higher, metabolic costs of locomotion. Of course, these costs are likely present, and animals may compensate for them, and they are difficult to measure. But if the costs are not detectable for one of the largest and most iconic sexually selected traits we know, we may deepen our understanding of these structures by looking at costs in a different way, not just by measuring their size, but through the lens of energetics. And with that, I'd like to thank a huge number of people who made this work possible, mentors, friends, and assistants, and thank you all for your attention.